From Microbe TV, this is Immune, episode number 2, recorded on November 15th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi.
Welcome back.
Thank you. I'm glad to be back.
What's the weather like up there?
It is very cloudy. We're entering into this time of year where it's kind of gray all the time, but it's not too cold. It was a little chilly this morning, but it's 41 Fahrenheit right now. So
it's the sad season.
It is.
You have a fluorescent light that you sit next to daily?
I have one at home. I don't use it that often. What I do instead is I try to find something fun to do in winter. So I take my kids skiing.
That's cool.
So that way, when I see flakes outside, I'm like, "Yay, it's snowing!" Instead of, "Oh my God."
Do you go to Greek Peak?
We do.
I remember that.
We do. It's very close by, and so it's small but cozy.
My roommate in college was a ski instructor there.
Yeah,
he was a Cornell student, and he did that, on weekends or whatever.
It's not a bad gig.
It's cloudy here too, 10 degrees Celsius. Also joining us from Worcester, Ohio, Steph Langel.
Hey now. How are you guys doing?
So Steph says, "Hey now" and "hello there." Those are the two things.
Those are the two. I might stick with the "hey now." I like that. It kind of
is that a Midwestern thing?
Well, I don't know. It's a comedian thing. There was a comedian I listened to that said that a lot, so that I've kind of adopted that, I think.
Cindy, I think Steph just can't bring her to call us by our first name, so she uses "hey now" or "hey there" or "hello there."
I'll get there. I'll have to work myself into it, I think.
We'll break her down.
Yeah, break those walls down.
All right, we have a couple of interesting snippets for you today. There is a neuroscience meeting going on in Washington. This is a big meeting.
30,000 neuroscientists from what I saw.
Why would you go to a meeting with so many people?
I don't know. It's huge.
I like 100 people. I think that's great because you can talk to everyone, but that's the way they do it. Also, there are lots of clinical meetings that are huge also. But a few interesting things coming out. One, I noticed, this is from Spectrum. They seem to be covering the meeting. Maternal immune response may render the brain vulnerable to injury.
Yeah, I thought this was very scary.
This is from mouse studies, right?
Right.
Correct. And it kind of comes off the heels of some work. I forget the journal, but showing that there is a boost of an autism risk for women who get potentially a viral infection during pregnancy. The experimental has been mice, but they have seen some evidence of this in humans.
Definitely, yeah. I saw that as well.
Yeah, so in this one, mouse brains exposed to inflammation in the womb become more susceptible to a second challenge. This is from the meeting. It's not published yet, obviously. And of course, when you're pregnant, you're also immunosuppressed, correct?
That is correct, yes.
So,
or at least you have an altered type of immune response.
Yes.
Yeah, they used to say that immunosuppression throughout pregnancy. I think now we're more thinking of it as different trimesters kind of have different features. So maybe not immunosuppression, but you're right, altered.
And then, so if this comes out at some point, something we could check out.
Yeah.
If they look at the details, because from the article we can't tell anything.
Yeah, not very many details, but I guess it fits also with why they tell women during pregnancy to get, for example, the flu vaccine. So it's a good reason to do that.
And if I remember, back in 2009 during the swine flu outbreak, pregnant women were getting very serious influenza.
Yeah.
Life-threatening, and that's part of it, I suppose, that altered immune response. And another article, also in Spectrum, maternal infection may trigger autism traits via neuronal patches. Patches of overactive neurons in the brains of mice exposed to inflammation may lead to autism-like features, a new study suggests.
It was interesting because, I mean, in these mice studies, they're looking at, okay, the mice born to mothers who got this hit of inflammation, this first hit, then if their babies got the second hit of inflammation, it seemed to be something to do with a gastrointestinal infection was more likely. And I looked and they said that there was a previous report that if the pregnant mice were treated with antibiotics, they were less likely for this autism-like features to happen, which, you know, we talk about antibiotic use as maybe a potentially bad thing in pregnant mothers, but maybe this could be, if this is true, more on the beneficial side. I don't know.
Treating the mice with an antibiotic ameliorated the issues there. Yeah.
Right.
Yeah.
Steph, I think you found something also in this RU Press JCB link. Is that right?
Yes, I am.
Oh, the Drosophila model of Fragile X.
Yeah, and it also has a maternal implication as well. And I chose it because I thought in other podcasts you do, you know, if you're trying to communicate science and you're telling people you work with flies, there's might not be a strong interest, and maybe politicians aren't really seeing the benefit of that. But they were showing that phagocytosis of innate immune cells can affect this Fragile X syndrome, which Fragile X, I'm not in autism research, obviously, but it's kind of associated with autism. It's a type of autism that manifests itself. So we can learn things from flies that we can apply to humans.
This article, Journal of Cell Biology, is from the laboratory of Mimi Shirasu-Hiza, who's my colleague here at Columbia. So what do you think if I get her on a future Immune and she can come here and we'll talk about this paper?
Oh, I think that would be great.
That would be fantastic.
Defects in phagocytosis. She's a big fan of our podcasting, so we'll see if she's willing to come on, right?
Yeah.
That's the true test if she's a fan, if she comes on.
Yeah,
That's right.
Some people are scared. So, Cindy, you found a toxoplate paper.
Well, what this was showing here was really interesting, and it was just another one of these spectrum stories. It was imaging studies linking immune genes to anxiety, so it sort of connects to the autism, behavioral risk things. But what they did was they started looking at mice that were deficient in various different immune genes, and what they showed was different patterns of activity in the brain in these mice that missed any of these genes. And so it's connecting the idea of behavior and the brain and the immune system and immunity and what these roles in these genes are doing. And I thought that was really fascinating. We probably need to look at it in more detail, but we could probably talk about that in a future episode as well. But it comes back to the idea that we know at least certain diseases are changing behavior. And one example that was shown is with toxoplasma. And I think you guys did talk about this on a TWIP at one point. But when mice are infected with toxoplasma, it changes their behavior and they seem to have a lower fear response. And so they've sort of connected this back and said, well, maybe that makes the mice more or less fearful of cats. And there's a definitive cycle between cats and mice to pass this parasite back and forth. And so if the mice then that are infected are less fearful of the cats, and then the cats catch the mice and then eat the mice, then they get infected and then they can spread the disease. It's just kind of an interesting connection of everything back together with immune genes regulating behavior and regulating infection. And I just thought the connection was really neat.
Yeah, it's cool. Just these few snippets make it really clear that immunology is really something that touches everybody's lives, right?
It touches everybody's lives and so many different disciplines of study.
Yeah, it's not just a matter of getting infected and being protected, but autism and other behavioral traits,
Alzheimer's, atherosclerosis, behavior, metabolism.
So we have to touch on all that, and that way we'll draw more listeners in.
Luckily, we have a lot of things.
Yeah, we have a lot of upcoming podcasts we have to dedicate to each of these.
Yeah, we only have 12 a year, though. It's going to be tough to choose, right?
I know. Well, that's true.
Okay. So that's an example, just one meeting, and these are all just reports out of the meeting, so we don't have any papers or anything, but lots of topics that are important for your lives. So stay tuned to Immune and we'll cover them. Of course, you can always tell us what you want to hear as well. We'll read some of our emails later. All right, we do have a paper for you today, and to tell you all about it, I'm going to hand it over to Steph. And I have to tell you, Steph, when I see your initials on the show notes, SNL, I always
what do you think of?
Saturday Night Live.
Yep, Saturday Night Live. Yep.
And I'm saying, why is SNL in our notes? People have told you that before?
Yeah. It used to just obviously be Stephanie Neal, but more recently SNL Langel. So I have gotten that a couple of times.
All right. This is a very cool paper. Tell us about it.
Yeah, I think it's cool as well. And it's pretty easy to follow, I felt. I thought about it. There was the most recent Nobel Prize. It was physiology or medicine. It went to 3 individuals. I'll list them. Jeffrey Hall, Michael Rosbash. I think that's how you pronounce it. You can correct me. Rosbash.
Yeah.
Rosbash. And Michael Young. 2 are from Brandeis and 1 is from Rockefeller. And they were awarded this because they discovered the molecular mechanisms of circadian rhythms. And some may have heard that before. But this was actually a paper published in January in Immunity, which is a Cell Press article, and the title, Lymphocytes, Circadian Clocks Control Lymph Node Trafficking and Adaptive Immune Responses. It's out of a German group at Ludwig, and I'll try to do my best German impression. I'm sure we'll get emails. Maximilians University, and it's David Drutz is the first author, and the last author, the lead author, is Christoph Scheierman. So we have 2 co-first authors, Steph. Oh, I apologize. Yeah, I hate to do that because that's that's a point of contention, I'm sure, in the lab as to who even gets the first. So let me not mistake. It's David Drutz and Olga Matvieva.
Right.
Are the 2 first authors. So sorry to Olga.
Steph, do you know what circadian means?
Basically, it means a rhythmicity that has to do with biological processes. And this could be something as simple as like night and day. So a circadian rhythm could be the lights come on, you feel awake. The lights go off, you feel tired.
Okay. And in Latin, circa diem is actually for about a day.
Oh,
circa.
That's kind of cool.
Yeah, right. Yeah, circa. Now, diem, because I know the saying carpe diem is seize the day, right?
Right, exactly.
That makes sense. So day, right. So that is one example, and that's related to melatonin and your pineal gland, but there's others. There's a lot of different circadian rhythms, and that's kind of what this paper wants to tackle, is looking at circadian rhythms or the cell intrinsic clock genes that produce proteins that help to turn a gene on or off depending on whatever that circadian rhythm is. So that's kind of the first concept of this paper. The second concept is lymphocyte migration. And basically, your lymphocytes are white blood cells that traffic throughout your body. They're circulating between blood to lymph node, they go back into lymph, and they do this because they really need to kind of survey your body for any type of antigens, and an antigen is just a piece of a pathogen, whether that be a virus or a bacteria or fungus, and they use they are able then to kind of become activated once they see this antigen or it's presented to them by another cell type we'll talk about probably in other episodes.
So Steph, so the lymph node is where the lymphocytes go to be shown antigens, right?
Yes, correct.
And they're shown by what we call antigen presenting cells. Makes sense, right?
Right.
So why don't they just stay in the lymph nodes all the time? What are they circulating for?
Right. Well, they circulate because they kind of have to deliver their message or live out their effector functions in the tissues where the infection is happening. Right. So they're going to go to the lymph node. They're going to be presented this antigen as kind of like an alarm system to say, hey, here's the antigen. This is what you need to know is going on. Develop a response to that particular antigen, traffic out of the lymph node into lymph, into circulation to then go to wherever the site of infection may be. And the effector function of that lymphocyte could be killing a virus-infected cell or producing antibody or helping to produce antibodies. So that's the basic function of a lymph node. And there's many lymph nodes throughout your body, in your gut, up by your clavicle, in your mammary tissue. They're all over.
And that's one of the reasons why we can tell when we're getting sick often, because they'll say, are your lymph nodes swollen, right? And it's because all of those cells went there. And when I teach this, it's like the nightclub where they all get together and talk, because you have the dendritic cells that pick up an antigen in one place and the T cells in a different place. And when you think about how many cells are in your body, and to get those two specific cells together in order to be able to talk is an absolutely phenomenal thing to happen. And so these cells go into the lymph node so that they can find each other, which, I mean, in the entire body, two cells to find each other it's an amazing feat that this actually works.
It is amazing. And I would love to kind of harken back. There was a TWIV, a This Week in Virology episode, I think in the 100s, Vincent, if you remember, like 2012, where you had, and I forget his name now.
Gabriel Victoria.
Gabriel Victoria, yes. And he is fantastic. Maybe we could have him on again if he'd be willing. He does a really excellent job of describing clonal production of B cells, and how exactly what Cindy was talking about, how that happens, how our bodies can recognize up to 1 trillion different antigens that we may come in contact with. So I divest. But
you are digesting, I hope.
Yeah, I'm digesting.
Digressing.
Digressing, yes. So yes, the lymph nodes. Now, the lymphocytes, they have to get there somehow, and there's a kind of a special mechanism by which they're able to do that. So I'll introduce something called a high endothelial venule. And these are important because these are endothelial venules that are basically blood vessels that are specially adapted for the lymphocytes to migrate to these secondary lymph nodes. And they're special because they're plumper than a normal venule that's going to be bringing nutrients or blood throughout your body. They're plumper. And so this is because the surface expression of certain receptors on the white blood cells, the lymphocytes, they kind of start rolling along these very plump endothelial cells on these high endothelial venules, or HEVs. And as they roll, if they're going to come in contact with some type of ligand or on the surface of the endothelial cell, they bind. And whether or not they bind with high or low affinity is dependent on the activation status of that lymphocyte. But it allows the lymphocyte to come in close contact with the venule, to then roll along it, be kind of drug into it, and then diapedesis through that endothelial venule and then into the lymph node. So it's a really neat kind of lock and key system where you have a receptor on the surface of a cell that's specific for a ligand on the surface of the venule, and they work together to get that lymphocyte to the lymph node.
Are these venules then are in they pass through the lymph node, right?
Right.
And so as these lymphocytes are passing through these, does every one end up in the lymph node, or only a fraction of them?
Only a fraction, and it can be dependent. I mean, at some point, the answer to your question would be yes, everyone would likely go through the lymph node because they do have to circulate to see if there's antigen to be presented. But not everyone is going to be activated or bind in a high affinity way, because if that was the case, we would have problems. We would have cells highly activated, migrating, and likely maybe developing some type of autoimmune disease or chronic activation status. So there's definitely kind of a checks and balances system with the way that our lymphocytes migrate throughout our body.
Okay.
So those are the two concepts. And, when I think about lymphocyte migration and what I was taught, I just assumed that these lymphocytes constantly migrate and circulate in kind of a linear way. I didn't really, when I envision it, I don't picture it being something that could behave in an oscillatory way or day by night changing. But that's kind of the presumption of this paper. There has been evidence that innate immune cells, they are in certain quantities at certain times of the day than others. And it bases off the premise, well, lymphocytes may migrate differently dependent on if it's day or night. So that was kind of an interesting concept to think about, that it's not linear. It could be different depending on the circumstance.
I think there's the other aspect that's really cool that I didn't know is that getting out of the lymph node is also regulated, right? What they call egress.
Yes, yes.
And there's a receptor involved. And this is the basis for treatment with some drugs for MS, multiple sclerosis.
Multiple sclerosis.
Oh, that's so cool.
Yeah, they stick all the cells and keep them in the lymph nodes so that they can't get out and enter the brain and cause the problems.
Because they're autoreactive cells, right?
Exactly. Right.
I just love that basic science interface with medicine, right? You figure
Absolutely. Yeah.
we have to emphasize that as much as we can. Here's people wanted to know what controls egress, and they figured it out. And then they translated it into a treatment for a disease. That's great. I just love it.
It is very cool. And that specific receptor, it's sphingosine 1-phosphate receptor. And so, as you said, if people didn't do work on this, you ask a graduate student, well, what are you working on? And they name one protein. A lay person might be like, you're working on one protein. That is, that's dense. That's very specific to one. How does that relate to me? And it does. And it's very translatable. So basic science, yes.
Sometimes we don't know how it's going to relate to us when we're studying it first, but then you read a paper or you hear something, you hear a presentation, and all of a sudden things connect together. So we just can never predict where the basic science is going to lead us.
Right.
Anyway, we digress again.
Yes, but but it's fun. It's fun.
Maybe the podcast should be called Digression.
Digression and egression, right? So anywho, that's kind of the assumption. So they hypothesize that there may be different migratory pathways of these lymphocytes through the lymph nodes in a circadian way. And the relevancy of that is, when we vaccinate, we are assuming that lymphocytes are in one place constantly. But really what this paper is going to show us is that may not be true, and time of day vaccination or treatment or prophylaxis may be something we should think about. So as I go into this paper, they kind of just wanted to define, how many cells are where at what time. They're doing this in mice, and they're doing this it's called Zeitgeber time. Now, again, German-speaking friends, feel free to email us. Basically, what I was looking up translation, it's just the timer, and a Zeitgeber time of 1 represents 1 hour after the light has been turned on. So we're looking at it in a progression of every 5 to 4 hours they have depicted here, and it just represents 12 hours of light and 12 hours of dark. And what do the cells do with those two different time points?
And this is because when we house mice in a facility with no windows, which is typical, you have a timer on the lights to give, unfortunately they come on in the morning and go off at night.
Which is actually opposite of what mice normally experience because they're nocturnal. So we experiment on them during the middle of their night, which is interesting.
Weird,
and given this this periodicity that Steph is going to describe, it makes us call into question when we do our experiments, how relevant is that to translating to humans?
Yeah, we do it for convenience, right? Because everyone comes in the morning, they want to work on their animals, right?
Right. And in our last podcast, we were talking about the temperature, and the temperature is set so it's comfortable for humans, not for mice. So it's really an interesting thought. Exercise there as to how we're treating these mice in these animal facilities and how realistic that is to representing what we see in human.
Right. So what they show is that they're looking at 3 different types of lymphocytes. I'll name them. It's CD4 and CD8 T cells and B cells, and they show them in circulation peaking at ZT or Zeitgeber time 5 hours after the lights come on. And they are at their lowest point in circulation about, hour 17. So during the light, when the animal is technically resting, they're at their highest. And when the animal is technically active during the night, they are at their lowest. And then that is actually an opposite of what you see in the lymph node. And so then they conclude that there's this oscillatory nature, which makes sense. You have the lymphocytes in your blood are peaking during the day, they're decreasing at night, whereas then you see them increasing in the lymph node at night. So they're kind of coming home at night almost, based on what they show here in mice.
And this night, again, for mice is when they would be out and about, right?
Yes, which is interesting.
And if they leave the lights off?
If they leave them off, it actually, it stays the same. So whatever that intrinsic, endogenous, whatever proteins are being produced to control this, they stay the same even with the lights off. Now, what's interesting is if you flip it, if you put the lights off for the first 12 hours and turn them on at night, which is opposite, you see the amount of cells homing or coming to the lymph node, it's opposite, which is interesting. It definitely says that light is what they call an entrainment factor. And I had to look that up, but an entrainment factor is basically something that it's what is able to turn on or change the dynamic of the circadian nature. So
that's interesting. So there's, in the absence of light, there's an endogenous timer still, or a clock.
Right.
But
right.
but they're clearly also responding to light.
Yes.
As you just said. Yeah.
Right. Right. So really interesting. And they show that in different types of lymph nodes, because obviously we don't just have one. There's many throughout our body, and it shows a similar trend where it's peaking during ZT time 5, decreasing at hour 17. For some lymph nodes, it's more than others, but generally the same. Then they want to prove, okay, well, it could be that the cells in the lymph node, maybe they're proliferating depending on night or day. Maybe it's not the cells migrating and leaving the lymph node. That's what is based on the circadian nature. Maybe the cells are just proliferating at a higher rate during at night rather. So what they did was they blocked the ability of the cells to migrate to the lymph node or to egress out of the lymph node. And if it was in fact proliferation, you would see the same thing happening. But what they saw is when you block homing means going, or egress, leaving, they see the cells either retain in the lymph node or they drop off. And that means either they're not getting there, so there's no cells there, or they can't leave, and so there's a huge amount of them there. So that helps to kind of show them that it is this circadian nature of how the cells are getting there and how they're leaving.
Now, do they show that this is a property of the cells themselves or a property of the lymph node that's creating this periodicity?
Yeah, well, I think how they went about that is they did adoptive transfer studies, and they so they took cells from mice that were housed at ZT5, which would be the time when the cells in the lymph node are at their lowest. And then they took cells from mice that were housed at ZT13 and they swapped them. Well, they first labeled them. They labeled them with CFSE, which is a dye, so that you can tell which cells are which when you're looking at them in the lymph node. But they basically flip-flopped them. And you would suspect that if, in fact, you took cells from a donor mouse that was housed at ZT5 and gave it to a mouse that was at ZT13, you would see the amount of cells in the lymph node go up, that it would match the cells that are kept at 13. But in fact, it's kind of halfway in between. It doesn't exhibit the full properties of the cells from the mouse at ZT13. So I think to answer your question, what that shows is cell intrinsic, but there's something about what's in the lymph node, a molecule or protein, that's also oscillatory, not allowing it to maybe exhibit what the recipient mouse would naturally have, if that makes sense.
Mm-hmm. And so the cells come to the lymph node because they're called there, right?
Yes, they are in a sense called there. It is kind of like, I like that they use the term homing because it is almost like coming home in a sense. This happens because of some of the things I talked about before. So you have these receptors on the surface of cells. I'll mention one that they focus on is CCR7. And CCR7 is expressed on the cell surface, and they see that this receptor is also exhibited in this oscillatory manner, peaking about the same time that the cells would be going to the lymph node. Well, that receptor has to bind to something. It can't just bind to nothing in order to get there. And one of the things it's going to bind to is its associative ligand CCL21. They found that CCL21 in the lymph node itself increases. It's kind of increasing just after you see CCR7 on lymphocytes increasing, which is an interesting story if you think about it, because you have the receptor on the cell increasing very nicely right after its receptor is increasing. So it's because of these pro-migratory factors calling the cells to the lymph node that seem to be acting also in the circadian way.
I love that. I think it's great that they're both cycling. So you can imagine that you could leave one on the lymphocyte, but for some reason that you have to have them both controlled, and maybe there's some negative aspect of having it on both all the time or on one. So
yeah,
it's pretty
right.
cool.
And there's a lot of other receptors and ligands that they could have looked at. Like I look at mucosal organs, and so I'm very focused on a surface receptor called alpha 4 beta 7, and its associative addressin or ligand is called MADCAM. And so there's a lot of different things they could have looked at, but I think for their purposes they looked at these because they saw that they had this oscillatory nature. You can really probably only focus on one lymph node. Otherwise, the paper would be too huge. But I would be interested to see what this looks like in the Peyer's patches, which are the lymph nodes of the gut.
Yeah.
I would love that. But,
in your mucosal surfaces, there's probably oscillation also, right?
I would think so, related to when you eat and things like that.
Yeah.
Which is fascinating when you think about how your microbiome changes dependent on time of day and how that can affect that does affect how cells migrate. So there's a lot to think about. But specifically with this paper, they do a couple different things where you want to prove that CCR7 is responsible. You're going to knock it down in a knockout mouse model. When you knock out CCR7, the oscillatory nature of its migration to the lymph node disappears. When you use something like pertussis toxin, when you treat the cells with this, it actually it effectively chemically disrupts the receptor. And so we don't see the oscillatory nature. So different ways that these authors removed these pro-migratory factors, and the trend goes away.
There's still the issue, though, that the CCR7 and the CCL21 have this periodicity and expression, but what regulates that?
Right. Well,
turtles all the way down.
Turtles all the way down. Yes. Well, that is a perfect transition into talking about the clock genes themselves. And these clock genes are going to be producing the proteins that essentially are regulating whatever is in question. And for us, it's CCR7 and CCL21. They looked at a bunch of different clock genes, but they really focused on this one called BMAL. Of course, I'm not in circadian clock research, so if it's not produced BMAL, if it's like BMAL, or you can let us know if I'm saying it wrong. But anywho, what they did was they decided to create knockout mice for this BMAL gene so that the circadian clock gene was not produced anymore. And in fact, when that happens, you have a decrease in CCR7 expression, and then you have a decrease in the ability of those cells to migrate at different times of the day.
I wonder what problems in general mice have when you take away this clock. I'm not familiar with that, but you would guess that there are other issues as well, right? Oh, look, it's necessary for fertility, BMAL.
Wow. So I bet if you tried to breed them without that's a good question, because we have to breed them in order to get knockout mice. So I don't know if you take a homogeneous BMAL mouse and bred it, would it actually go forth with fertility? I don't know.
If you wanted a homozygous, you could breed, heterozygous parents and just take the offspring for your experiments.
True.
Right?
But it'd be interesting to see if you took those offspring and tried to breed them.
Yeah, sometimes it's male or female specific as well. I don't know in this case. Sometimes you can breed the homozygous male to the heterozygous female or vice versa to generate more of the knockout pups.
Sure. So they are able to prove so they identified BMAL, they knocked it down, they saw that the oscillatory nature disappears, and that is dependent on CCR7. So they really nicely show us that homing to the lymph node is in fact dependent on these clock genes. So what happens then about egress? Is the same thing seen if we are able, if we knock down these clock genes and then we look at some receptor responsible, like we talked about earlier, sphingosine receptor, if they're able to then exit or egress rather. And so similarly, when they look at that, circadian oscillations, the lymphocyte egress from the lymph node, when you knock down BMAL, you do see the oscillatory nature again disappear. So it's not only the cells migrating to, but it's the cells leaving that are both affected by these circadian clock genes. So there is a second part of this paper about adaptive immune responses, which I will dive into, unless Cindy and Vincent, any other comments about kind of the immunological part looking just at lymphocyte migration?
No, I think that pretty well covered it for me. I think the exciting part of this is what you're going to go into now, which is how does this impact immunity, right? So we know these clock genes are there. They're regulating expression of the ability of these cells to migrate into and out of the lymph node. And we said that meeting up with the antigen presenting cells in the lymph node is so critical for activating these immune responses. So what happens when you mess with this whole system?
Right. So what they do is they present a couple of models of ways to show us of how disease can be affected by this trend. And the first thing they focus on is a model called EAE, or experimental autoimmune encephalomyelitis. I think also it can be called, I've heard it experimental allergic. Instead of autoimmune. I've heard both. But basically it's modeling I'm not thinking
multiple sclerosis.
Yes, sorry. Thank you. Multiple sclerosis, or basically brain inflammation. And so what they do is they inject animals with pieces of small pieces of probably the CNS.
I think it's the myelin sheath that covers around each of the neurons. And so if those so what happens is the neurons are surrounded by this myelin sheath. And if the immune cells recognize that myelin sheath, they start to attack it. Mm-hmm. And when that myelin sheath is disrupted, the neurons can no longer transmit their signals, and you end up with this paralysis.
Right. And so what they demonstrated was that if you immunize with these mice at different times, they actually have worse or less clinical scores. So they've looked at ZT8 and ZT20. So the animals that were immunized at ZT20, they actually experienced less clinical scores than the ones that were immunized at ZT8. And ZT8, remember, is when the lymphocytes are actually, they are more in the blood than they are in the lymph node. And then that is flipped at ZT20. So it just demonstrates that for something autoimmune, that this potentially, depending on, I don't know, when they start exhibiting symptoms, it can be different depending on the time of the day.
So this means if you have MS, you should just stay in the dark all the time?
Well, no, because remember, if you keep them in the dark, they maintain their
the cycle.
Yeah.
Exactly. And, when we think about this being a mouse model, it's going to be different. They're active at different times. And so I like applying what we're seeing here in this paper to real life, but thinking about the differences in species is also important.
Do we know if has anyone ever looked in people? It'd be easy to look at blood lymphocytes at different times of day. Has that been done?
Well, we looked at that a little bit in the last episode. We didn't look at CD4 and CD8 cells, but we looked at monocytes, and there was a time of year dependent change. But they've also looked at monocytes by time of day as well.
Right. So yes, I would say yes, they have looked at that, and it does fluctuate. And they went on then, they looked at this. I have to say, one thing about this paper is they squeezed what I find, and maybe what we all find to be some of the most interesting stuff. In that paper, it represents 15 lines of text and one graph. I wish that maybe it was split more down the middle, because they really rush in. So within two graphs, they present to us an influenza model where they infected mice with influenza, and also a Helicobacter model where they look at Helicobacter infection. Now, I will start with the influenza, and what they saw was that depending on what time of day that they were infected, they had a difference in the amount of, I think it was interferon secreted from CD4 T cells rather. Mm-hmm. And so showing that depending on when you get sick, it could matter for how your cells are working to fight off that infection. Now, the Helicobacter one, if I'm looking at this correctly, they don't show differences between giving Helicobacter at two different times. They just show linearly it increasing and then decreasing. So I'm not so convinced about the Helicobacter one. What do you all think about that?
Yeah.
It doesn't actually have a control there.
Yeah, that's correct. They infect them, and then lymph node counts 3 weeks later
That's right.
But that would happen normally in an infection.
Yeah.
You would see that lymph nodes or lymphocytes increasing. So, Helicobacter, I'm not so convinced. But the influenza, I would have liked to seen more than just CD4 T cell data. But it does show that depending on a time of day, the mice had different responses. So overall, I think this was a really interesting paper, relatively straightforward and understandable. And I think it does have some implications as to when we vaccinate, when we give treatments. My pick of the week is actually looking at people who have had wounds or burns, and how they heal is dependent on time of day. So it's really has some interesting implications. And I'm glad that kind of the Nobel Prize brought more of this to light, looking at these circadian clock genes.
Obviously, you could have many, many papers on infection. And looking at these, I would have liked to see, for example, the influenza infection done in the clock mutant mice, and the mice lacking the chemokines receptors and ligands, and the egress receptor, etc. That would be very interesting to see.
Exactly. And I don't know, you have to wonder, were those experiments something that were asked of the authors before publishing?
Yeah.
I don't know, but
Maybe.
I thought to include adaptive immune responses in the title, to me, it would be nice to have that be a bit more robust.
Yeah.
But,
I think
future
that's
experiments.
Correct. Yeah. I mean, most of it focuses on migration, right?
Right.
In the absence, and there's just a little bit on immune responses. So.
Right.
So I thought it was interesting. They in their discussion they quote a couple of studies with vaccination in humans.
Yes.
Recent studies of hepatitis A and flu vaccines in the morning yielded highest antibody titers compared to other times. So that's interesting. There are 2 papers there, and I don't know who gets immunized in the morning. It's usually in the afternoon. And I don't know if anyone gets immunized at night, right? That would be a problem, I guess.
Right.
Maybe it would lead to lower antibody titers. But I guess that's one. So, Steph, if you had to tell the person on the street why it's important for them that lymphocytes traffic in a circadian way, what would you tell them?
Well, I definitely would say that it could have some real life effects as to when you take medicines, when you get vaccinated. I mean, like you said, the reality of it is we have office schedules. We can't always optimize this based on what we're seeing here, but I would assume, especially for maybe some really high risk patients, like people who have multiple sclerosis or people who it's a life or death situation, if you could respond better to a certain treatment by giving it in the middle of the night or at an odd hour, I think that is important.
I think what's interesting is to consider, there's a sizable population that works at night.
Yes.
Right? If you take an overnight flight to Europe or wherever, you're in a plane with lots of other people breathing air and with viruses in it. And you can imagine that, you're a little more susceptible maybe in those hours, or you're not going to respond.
That's what I was thinking about. We think about people saying, I went on a flight, I got sick. But you wonder, is it because there was an exposure, or was it because there was an exposure at a certain point in time when they were more susceptible?
You could certainly think of doing those kinds of studies, right? Just a descriptive study. People on airplanes, is there a difference in their immune responses, as they get infected during the night? So I think those are cool things to think about, right?
Definitely. And maybe even easier that you could control better in terms of a epidemiological study was looking at people who work night shifts and if they have lower responses to vaccines or whatever than those who don't. So I do think I like the paper. I thought it was really interesting and it was good for our podcast.
I think one thing you might mention also, they say our data show not only do T cells migrate in a circadian manner, but also dendritic cells, the major antigen presenters, right?
Exactly.
Yeah.
So that makes perfect sense, right?
Yeah. If one was on one periodicity and the other one was on a different, that would be bad.
That would be bad.
Got to coordinate,
right? And you could do that experiment. Actually, you could probably mess with as clocks in a cell type specific way and see the effect of that, right?
Exactly. You could knock it out in a T cell or a dendritic cell. And I believe they did some studies with T cell specific knockout, and they were able to show some intrinsic activity as well.
Yes, they did. Yep.
So this for me is totally new. I didn't realize this at all, that the lymphocytes moved in this way, but it makes you think, what other things, depend on circadian rhythms besides, there are all kinds of things you could think about, and whatever you're working on, if you happen to be working on something, you may want to start thinking about it in a clock manner, right?
Right. And if you are near a computer, but of course don't get on your phone, but when you are at one, there's a really neat visual that they have. I don't know if you guys have played that, where basically they take their entire paper and they summarize it into this pictorial, and you can press play and they show you what's happening to the cells as they enter the lymph node during the day hours, and then it transitions into night. So it's a nice, if you don't have time to read the paper, just press play and you'll get the gist of what we're talking about.
Did you say that this was open access?
It is open access, which is
that's
great.
Good. That's very good. So you should go take a look at that, because that's a feature that is kind of makes it a little more accessible.
Yeah, for sure.
All right. Well, that's great. I love that. Good stuff. And I have to say that if you, there's a wonderful book by Jonathan Weiner called Time, Love, Memory.
Yes.
Yeah.
Written in 1999, where a few and Time, Love, Memories refers to Seymour Benzer's work to identify genes that control time or clocks, love, mating behavior in Drosophila, and memory. And so he did all three, and there are a number of chapters that focus on the clock, and he talks about Rosbash, Young, and who's the third individual who got the Nobel?
Rosbash, Young. Oh gosh, let me go back.
Hall.
Yeah.
And their stories from start to finish. It's just great. And he's a professor here at Columbia, and I emailed him and I said, "What did you think?" And he said, "Oh, I couldn't believe when I heard it." He's, can you imagine writing this story and then 20 years later they get the Nobel Prize? How good that must make you feel as an author. So I highly recommend that book. It's really, really good.
To put that on my list.
Hall was a postdoc with Benzer at Caltech. And actually, the first clock mutant was made by another postdoc, Konopka, who did it in flies, and it was a really arduous procedure. He never got tenured, unfortunately, and left, and of course didn't get the Nobel Prize. Hall took the gene and moved to Brandeis and was working on it, and he met Rosbash, who was a molecular biologist, so they collaborated in cloning the first clock gene, and then at the same time, Michael Young at Rockefeller was doing it independently. So it's a great story, totally wonderful. So this is a great paper to do now because it's timely with the prize.
Yes, right.
Well, we do have some email. If you're finished, Steph.
That's it for me, unless anybody has anything else.
Yeah,
but
that's good. That's good.
Cool.
Let's go through a few. We're lucky. We have a bunch of email already.
Love the email.
I know. After one episode, that's fantastic.
Keep them coming.
So let's go through these. We'll take them in. Take turns. The first one is Kimia, who writes, "I'm looking forward to the immunology podcast. Just wanted to say that I would like to hear about immune responses in response to viral infection. It would be nice also if the podcast could focus more on human-based research." And Kimia is a PhD student at the Karolinska Institute, which is where the Nobels come out of, right?
Yes. Right.
Of course, we will. My presence on this show will guarantee that we will look at immune responses to viral infections. I'm sure Steph would also.
Our next paper, if we are doing the Zika paper for our episode 3, it applies to both those things, viral and human.
Yeah.
That's right.
Steph, you want to take the next one?
Sure. Gavin writes, "Dear tycoons of the immune." I like that. "I'm writing in for my chance to be the first ever emailer. I think he may have been the second, but close.
Yeah, he was second.
To the new podcast. I studied virology in grad school, but my PI kept sending me to immunology conferences, and I took a shine to the subject. I had the opportunity to listen to so many rock stars at Immunology 2016. I'm confident that you'll never run out of amazing guests." Very true. Stephanie asked for recommendations on Twitter, and I thought it would be cool if you could occasionally bring in a physician specializing in allergy and immunology. Dr. Griffin adds a lot to the value to TWIP, which is another podcast Vincent does, one of the many this week in parasitism. I picked up a copy of Janeway's case studies in immunology, and it seems like there's a great opportunity to borrow the case guess format. Thanks again for all you do. And that's Gavin. And I agree. I think I'd love to have physicians on to talk about some of the clinical applications of what we talk about.
Yeah, we'll work on it. Gavin's a medical student at UCSF. Wow, that's great. Cindy, you want to take the next? You can take the next two, actually.
Great.
Got two big ones.
So Trudy writes, Dear Immune Trifecta, I can't even express how excited I am about this podcast. I have waited for an immunology podcast for about 7 years. That's a very long time. Ever since I started listening to TWIV, TWIM, and TWIP. I can already tell that this will be the type of podcast where I will repeatedly rewind certain portions just so that the information will stick. I already learned so much from just the first show. In response to your request for suggestions, could you please talk about the complement system? Thank you so much, Trudy. So before I read the PS, it's funny that she wrote the complement system, and I commented on this when we were talking to each other in chat. That I said, really, most students hate the complement system because it's so complicated.
And then, well, if we do the complement episode, we'll have to have complicated complement or something in the title.
Yeah, that would be cute.
Yeah, and we should probably have a person who knows it well, because it is a lot of different moving parts there. So, but incredibly important for our immune system. Defects in complement make for very, very bad problems. So Trudy also says, P.S. Stephanie, I know exactly how you feel right now with your hopefully imminent graduation. I was exactly the same place about 10 years ago, and I remember the stresses and frustrations very well. Hang in there. You will do great.
Oh, Trudy, that's very sweet. And actually, that's very needed at this time. So I appreciate everyone's positivity. That's nice.
Yeah, you get towards that end and you start generating a lot of data really quickly, but you start having this panic feel, am I going to be done? And if I am going to be done, what am I going to do with this?
Yes, it's a lot of that. Yes. So I'm glad that there's a lot of people, obviously you and others, who've gone through it. So I can glean from your experiences.
Yeah. So the next one is Niraj. Niraj writes, Hi, Vincent, Cindy, and Steph. Following the recommendation of the podcast in eliminating the designation bias. That's good. First and foremost, absolutely thrilled and excited about this new podcast series, which, to be honest, I have been eagerly waiting for. And incidentally, who better than the father of the Twixes, Dr. Racaniello, to embark upon it? Listening to the first episode, I was taken down memory lane quite a few times. Having spent a memorable year at Cornell's chemistry department as part of the Tri-Institutional Chemical Biology, or TPCB, program, I had the pleasure of enjoying the most beautiful fall and the ordeal of facing the most spine-chilling cold winters within the same year. Ithaca is gorgeous. I'm sure Dr. Leifer can relate to this. And amazingly gorgeous. I missed the trips by the Ithaca Lake. And I'm gonna interrupt this reading of this for just a second. So those of you who laughed got this Ithaca is gorgeous probably because you've been here. So Ithaca is an area that was geologically formed by glaciers, and so we have a lot of gorges, and so they create incredibly beautiful waterfalls. So it's an amazing outdoor environment. And if you ever have an opportunity to come visit, you should. It's just absolutely gorgeous. But we say Ithaca is gorgeous versus gorgeous. And there's a play on words there because, we have these gorgeous. Anyway, continuing on. And finally, having a graduate student in Steph as part of this trifecta is just such an interesting blend to have for a podcast. And I'm sure it will be immensely rewarding experience for everyone. The world of immunology is so dense at times, terminologically chaotic, that it helps to have experts delve into and enlighten the broader audience. And I must say, it was fun to discover how the toll in the TLRs came to be. Who knew? Personally, having done a postdoc at Genentech in an immunology-heavy work environment, I've certainly had my fair share of momentous experiences where discovery just makes you sit in awe and be inspired. Thus, in line with this, I'm attaching a link to a paper by a former colleague at Genentech, and we can put this in the show notes. But what Gavin is talking about here, Niraj is talking about here, is that this was a remarkable and time that he was there at Genentech. So it's because it was one of the most seminal discoveries in the world of innate immunity that has probably seen in recent history. I'm obviously biased since I come from the same group. Given that no Nobel Prize has already been given for the discovery of TLR4, which, by the way, actually twice, once for Toll and once for TLRs, being the cell surface receptor for LPS, this study for the first time showed that in addition to TLR4, there's an intracellular sensor called caspase-11, which is highly conserved aspartyl protease. And mice challenged with LPS with a TLR4 knockout genotype, meaning they don't have any TLR4, survive only if caspase-11 is present and not otherwise. And this was groundbreaking. Historically, it was always thought that caspase-1 was a downstream caspase that activates pyroptosis or pyroptotic cell death. But my colleague for the first time discovered that for LPS, this is actually caspase-11 is more critical. And I'm going to skip over a little bit here, but this is actually a really interesting story because when they found out that there was this caspase-1 knockout mice that they were working with also had a mutation in caspase-11 and was in fact a double knockout, people freaked out a bit because they had defined a lot of biology around this caspase-1 gene, and it turns out some of that was due to caspase-11 and not caspase-1. But this was from Vishva Dixit's group at Genentech, who's an incredible immunologist. But many different groups have shown that this is in fact true, this caspase-1 versus caspase-11 phenotype. Including one that Niraj mentions, Dr. Shao. And so he says that follow-up studies looked at a downstream effector called Gasdermin D, which assembles to form membrane pores leading to spillage of cellular contents and eventual death. So at some point we should talk about the different kinds of death because there's a lot of different kinds of death in immunology. There's apoptosis, pyroptosis, necroptosis, et cetera, et cetera. So it's kind of interesting. But anyway, Niraj ends with, personally for me, this was a totally remarkable phase, even though I wasn't directly involved in the research, as I could see from close quarters the excitement and hard work that went into making a totally transformative discovery. So I just wanted to suggest one or all of these studies as food for thought and possible discussion point in a future episode if you deem feasible. I think it's a great idea. Overall, I would like to personally thank all of the folks who have been willing to have devoted their time to this podcast. And as a fellow former graduate student, I totally empathize with Steph and her wish to wish her the very best in completing her PhD without asking the dreaded when. And and to put things into perspective, here's a cartoon from PhD Comics. This is great.
I love it.
I'll just describe this really quickly. So this is a graduate student, frantic graduate student, talking on the phone with her mom. It says, "Mom, I'm trying to finish my PhD. Having kids is the last thing on my mind." The mom says, okay. What about after? And the student responds, "Well, I might do a postdoc after this." And so the mom says, "And after that?" "Oh, I don't know. Another postdoc? Then tenure takes at least 6 years." The mom says, "Your thesis is the closest I'll get to a grandchild, isn't it?" And she says, "Yes, and you're interrupting Leifer."
Great.
It's like that comic was on the phone with me and my mom like a week ago.
You should send it to your mom.
I will.
Do you think she'll appreciate it?
Yeah, she'll get a kick out of it.
She has to have a sense of humor, obviously.
Yeah.
All right. We have one from Ben. "Hello, Immuners. Congratulations on the new show. Really excited to learn more about the immune system with this podcast. With regards to suggestions for what you might cover, I'd love to hear some discussion of philosophical implications of the field of immunology. Of course, I realize that scientists are often reluctant to discuss questions beyond what the data shows, but I hope you might agree that addressing some of the larger questions that immunology raises might help to add a human dimension to what is undoubtedly a very complex subject." He sends a link to a paper that he's recently read, "The Immune System and Its Ecology," from the journal Philosophy of Science, which I think can be read as a really interesting argument for why our conceptions of immune function have really important philosophical implications. Perhaps you could discuss this paper in a future episode, or at least share a few thoughts on the relationship between immunology and the philosophy of science. So this is looks like it's a blog post, The Immune System and Its Ecology by Alfred Tauber, which was published in 2008 in this Philosophy of Science originally. Now it's been reposted on a blog, I guess. Mm-hmm. And, philosophy and science used to be the same field, right? Many years ago. Yeah. And they separated when science developed as an experimental framework. So it still has many roots in philosophy. And I think these are interesting topics. I would love to get someone on who, thinks about this all the time, because most, as Ben points out, most working scientists don't really think about these kinds of things. Maybe later in your career you do, when you're not doing as much science, because it's really intriguing to think about how these
things
things
and
align.
it takes a lot of brain energy, and it's not something that we do every day, for sure.
Yep. And I was going to say there was one philosophy of science class that I took that was offered. And you're correct. It was one of the harder classes that I've taken, because it really, the way that you were forced to think was so outside of the experimental way that we think of in a lab. And it was really rewarding, but it was definitely challenging. So, and the person who taught it here is a entomologist, so he's excellent. So maybe he could be a good person to.
Sure.
So we actually read that. So we read Tauber's The Immune System and Ecology, and it's dense. But what's great about it is it does put immunology and the basic science in the perspective of like systems biology and the environment, and it's great. So yeah, it'd be great to talk more about that.
I had a pick this week on Tweevo, which was an article in eLife called "How Should Novelty Be Valued in Science?" And I had an interesting quote, which I want to read because it's kind of relevant. It says, "Science as we practice it today is a relatively recent development. Our system of peer review, the priority rule, and the organization of scientists into cooperative social teams that compete against other groups of scientists all trace their origin to decisions made by the Royal Society in the late 1600s. For most of history, humans acquired knowledge outside of what we would recognize as a scientific framework. It would be unwise to assume that science is a permanent feature of our society, or that it can withstand deep structural changes and remain an efficient engine of discovery." So I think that's really important. Science, as we know it, us three and our colleagues, is really a very new thing, and it's likely in flux. It's not going to stay the same forever.
Yeah.
Okay, we have one more, and I'll hand this over to you, Cindy.
Okay, this last one says, "Dear Immune Team, I love your new podcast. Keep it up." Oh, this is from Roman. In the first episode, you mentioned that some macrophages originate in the yolk sac during development. Do these macrophages survive in an adult organism? I imagine that each individual macrophage cannot live for years, but they may multiply by division, preserving the line. Do these yolk sac macrophages occupy any specific tissues, or are somehow different from the bone marrow ones? I'll address that before we go on to the next part of his question here. So I did mention yolk sac, and I went and I looked at this a lot more and realized that I didn't know as much as I thought I knew about this whole business. But yes, in fact, there is a wave of tissue macrophages that originate from yolk sac and are independent of the hematopoietic stem cell, which is shocking to a lot of people who think about how the immune system develops. We thought that there's a hematopoietic stem cell that gives rise to all the lymphocytes and immune cells that are in the body. But this first wave is from the yolk sac. And in fact, what I found out is that it seeds all of the tissues and then they disappear. And a second wave comes. The ones that don't disappear, though, that remain are the microglial cells. So they do seed from this original yolk sac, and they remain. And so the other interesting thing about this is that we thought for a very long time that macrophages were what are called terminally differentiated cells, that they don't divide again. And what has come out over the last 5 to 10 years is the fact that macrophages do in fact proliferate in tissues. And so the idea that some of the tissue macrophages, including the microglial cells, derive from a non-hematopoietic precursor and can proliferate in tissues makes you question sort of what really is an immune cell? Where do we define what's an immune cell and what's not an immune cell? It's kind of interesting. But yeah, so there's a paper published in Nature that we can put in the show notes by Gomez. I'm going to butcher this name, Perdiguero. In Nature in 2015, that's called Tissue Resident Macrophages Originate from Yolk Sac-Derived Erythromyeloid Progenitors. And they do some really elegant work there using some fate mapping techniques that we can talk about on a future podcast, that basically at a specific point in time, you mark cells and all of their progeny with a fluorescent marker so you can tell who is derived from a particular cell. And the second paper is by Hashimoto, and it was published in Immunity in 2013, and that one's called Tissue Resident Macrophages Self-Maintain Locally Throughout Adult Life with Minimal Contribution from Circulating Monocytes. So we thought that there were these tissue macrophages that were present, but that a lot of what goes on in the tissue during infection is because of the infiltration of circulating monocytes that migrate into the tissue and differentiate into macrophages. But these two papers really question that. So the idea that some of these are non-hematopoietically derived and some and can proliferate in the tissues during an infection and are a major contributor to the host defense. And so it really, a lot of this work over the last 5 to 10 years has really questioned what we think about, what we thought we knew about macrophages and what they do in biology and how they derive.
And I think that's why immunology can seem pretty daunting to people, because not only are there so many things to define, but it does change. I mean, the concepts change quite drastically over, like 5 to 10 years. So I love this, what you just said, I find fascinating, and I did a deep dive. It's kind of going to be my weekend reading.
It did keep me busy. They're very dense papers, and they just have these amazing techniques. Immunology has some really creative ways of analyzing things. You delved into a little bit of that in the paper today with looking at cell-specific knockout of genes, but we have cell-specific knockout, cell-specific expression, time-dependent knockout, tissue-dependent knockout. There's just so many different variations of this can just lead to some fascinating observations.
Did you do the second part, the probiotics?
Oh, I forgot that part. Yeah, right. So Roman asks also, and another question about macrophages, if you don't mind. My wife is giving probiotics to our son, and it occurred to me, how do macrophages know which bacteria are good and which bacteria are bad? Well, this is a big question, but it seems that context matters. So good bacteria pretty much mind their own business, and they stay put in the gut lumen for the most part. And bad bacteria break through the barrier and enter into the tissue and induce an inflammatory response and induce immunity. That being said, we know that there are immune responses to both good and bad bacteria. So we have, for example, antibodies to components of microbes that we don't consider bad bacteria that are part of our normal microbiome. And so there's this whole idea that good bacteria are controlled by the immune system with a lot of antibodies and other mechanisms, but the bad bacteria gain entry and then they cause problems. And so the real answer is the macrophages may not know the difference per se, whether they're seeing a good bacterium or a bad bacterium, but they may really only come into contact with the bad bacterium when there are breaches in the epithelial layer, and that is likely part of what initiates the whole response. And so there's some data on different toll-like receptors, which is my passion there, from probiotics that are less potent. And so when you take probiotics, they might actually be blocking some of the inflammatory signals that you might get from some of the more stimulatory ligands.
Yeah, there's a lot of work in this area. We'll definitely be touching it.
Yeah, it would be a good episode.
Yeah, for sure.
Yeah.
Thanks for that, Cindy.
Sure.
All right, let's wrap up with some picks of it should be picks of the month, right? Because we're a monthly podcast. Right. Steph, what do you have?
Yeah, I originally had chosen something differently, but I think due to the timeliness of it, I'm going to talk about the grad student tax for just a hot second. So basically, and feel free to fill in if I'm missing a piece of this, but in the proposed tax plan that has passed the House and is now to the Senate, graduate students will be taxed upon the graduation credit that they receive on their education. So for instance, grad students, they get if they're working for a lab that pays for both, you're going to get your classes paid for. So that would be tuition. And you're also going to get a stipend. And in my experience, that stipend is it's meant to get you by. You're going to pay your rent. You're going to buy food, medical bills. And there's not anything left after that. And maybe maybe you go into the negative depending on how you budget. But what this is proposing is that we are taxed on the tuition that we get a credit for. And why this is a problem is because that tax would be more than what we make in our stipend. So then you're basically telling graduate students who are trying to become our future scientists and innovators and entrepreneurs that you can't afford graduate school anymore unless you have some form of support system. So this plan would be devastating to graduate students. People would have to drop out. They would have to find alternate potentially careers, or it would just be then that people who had outside support systems, meaning are people maybe from not diverse backgrounds, lower class, and socioeconomically, cannot go to graduate school. So I called my senator yesterday, who does support this, Senator Rob Portman, called him, left a couple of voicemails. I'll call again. He'll be sick of hearing from me. But I would encourage those. And I don't want to call this is very specific, dependent on the party, but I'm not going to focus on that. What I want to focus on is that this will disrupt our ability to really be leaders in the forefront of science and innovation, and how we all stay healthy and we combat things like climate change and agriculture. And so looking at this very personally, I could not afford graduate school if this went into play. And I go to a public university. So I think about the students who are going to Yale and Columbia and Cornell, whose tuitions, the credits they get for that tuition is much higher than mine. So it's very serious, and it would be devastating. So I implore people to call their senators and tell them to not support this.
Also, this would also apply to undergraduate tuition. If you are getting tuition in any form assistance, you would pay tax on it.
That's
correct.
I
heard a podcast yesterday about a janitor who put his 5 kids through college working as a janitor, and he would not be able to do that now because he would be taxed on the tuition assistance he got. This is totally absurd. The bill is horrible in all ways. It enriches the rich and takes away from the middle class. And as you know, the House stuck in at the last minute, taking out the mandatory subscription to Obamacare.
Right.
And you may ask, well, why would they do that? Well, it turns out there's a $300 billion pool associated with that, and they need that to pay for the tax breaks that corporations and the rich are getting out of this bill. This bill is horrible. There is just no way that any person of sound mind can support this bill. And everyone needs to realize it. And as Steph says, it's not about political parties. It's just about treating people properly. You need to call your senators over and over again and tell them not to support this. It's horrible.
Yes,
I agree.
So my pick is a New York Times article that talks more about this if you're interested in that. Yeah.
It's a very it's a thorny point here.
No, it's good. I mean, and the thing is they are on break right now for a week for Thanksgiving. So flood their voicemails with calls, because then they will hear them. And I don't know the date when they will vote on it. I think it's like the first week in December. So yes, flood the voicemails, email them. It's very important. And, we have data that shows that our ability to educate different classes of people who are not typically seen in professional spaces, they need to be there. They matter. And this bill would kill the ability of those of people who are not wealthy already to go to school.
I don't know why we have to enrich those who are already wealthy by taking away estate taxes and other taxes. And one of the provisions of this bill is to keep golf courses tax exempt. Golf courses. Why? I don't know. I just don't understand why you have to penalize people. This country has always been about taking care of people. And I think this bill just doesn't do that. And unfortunately, if you look at the ads on TV, they're very misleading. You'll have people saying, I support the American people. I'm for this tax bill. And people are going to look at it and go, okay, that makes sense. But it doesn't make sense because you're not getting beyond the fluff. So please don't be fooled. Of course, our listeners are already understanding that this is a bad bill.
Exactly.
Right.
I like two things. Oh, I was going to say, I like two things, Stephanie, that you said. One, you said it's not so much about party lines. It's about thinking about the future of science.
Totally.
And that's what's really important. Keeping that in mind, regardless of what your party affiliations are. The second thing you said was you picked up the phone. And I think that is a really big deal because there's a lot of email blasts out there that encourage you to click on this and fill out the form and put your name, and it automatically sends to the senators. And I have talked to our representatives, and they say that those are just basically, that's junk mail. Yeah, but if you pick up the phone, somebody at one point will either listen to the message or they will answer the phone. And if you can get somebody on the phone and they have to write down a note, they are supposed to share that information with that representative or that senator. And so taking the time to try and call as opposed to send an email or a snail mail letter, I think is the key. Right?
Absolutely. I hear that over and over again. By the way, I heard a wonderful interview with Tammy Duckworth, who is senator from Illinois, and she is so eloquent. I don't know if anyone knows her, but she used to be a helicopter pilot.
Wow.
And
didn't know that.
Became a commander, and now she's a senator, and she just says this is a horrible bill, and it doesn't help people at all. It helps big businesses, and she says what we need are mom and pop businesses given help, and that doesn't help them at all. So, everyone is busy, but you have to get engaged. And she said call, and that's really the thing to do. All right, Cindy, what do you have for us?
Well, I don't want to continue a negative trend, and I certainly can't follow that. But I read an interesting paper that was in PLOS One, and so it's open access, and it's about men asking more questions at scientific conferences. Now, this was a limited study, so it has its own caveats, and it was only one conference. But the analysis showed that men ask 2/3 of all the questions, and you might say, why is that important? Well, there were actually, when they did the counts, there were 50% of the audience was women, but 2/3 of the questions were asked by men. And part of asking questions is challenging the people who are giving the talks. It's showing your knowledge within the community. It's engaging. And so when women aren't doing that, they're not being recognized as much. And one of the things that they did was they said, okay, well, maybe it's the diversity of the chair or the speaker. Because maybe if it was a woman speaker, more women will be likely to ask the question. Or maybe if the chair of the session was a woman, they'll choose women more often. But the end result of their analysis was that didn't actually make a difference. It didn't matter whether the speaker was a woman or a man. There was still this disparate question asking by male and females. And in fact, they only had out of, 270 total questions in 31 sessions, which when they looked at the analysis, I thought that was kind of on the low side, which we'll wonder whether we're spending enough time asking questions at conferences these days. But they had only 36 instances where there was more than one person who wanted to ask a question at the same time, and they didn't see any preference. So the chair didn't choose a man over a woman, but the numbers were really small. But I think that what this tells us is that probably a larger analysis is needed, and we need to think about stepping up and asking questions more. And I also think giving more time for questions at seminars and at conferences. I know my experience going to a recent conference, so many of the speakers tried to cram in so much information into one talk that they went incredibly over in their time every single time. And so the question and answer part of it got cut short every single time. And I think this is a disservice to the scientific community, because that's an opportunity for people to interact, to spark new ideas, to have a discussion over the data that are presented. And I think we're missing out these days in the way the conferences are designed and the way the speakers are choosing to put too much in and go over their time.
Yeah, I totally agree. I think the questions are the best part. And there are two reasons for going over. One, you're not properly prepared. you haven't rehearsed enough so that you nail it on the time. And you can tell some people do and don't, others don't. And second, the chair has to say, look, I'm going to get up at 1 minute and you're going to stop at 2 minutes. And you have to do that. You have to enforce it. And most people are afraid to, but you have to. Otherwise, you miss out on the best part, which is the questions. Now, this is really interesting. However, I suspect that, as you said, we need to look at other conferences because it could be there's a big variation that occurs, right? Over
sure, absolutely.
Because I've certainly been at sessions where lots of women ask questions, maybe more than men. So if it's true in overall, then it's bad. And I think we have to start at the grad student level and encourage people to ask questions, because if you get comfortable with it in your local environment, then you're more likely to ask questions outside of it.
And this could be dependent too upon, in biology and in biomedical sciences, I do see more, and maybe an equal amount, or at least more women than other fields. For instance, if you're in like physics or engineering, where there's a lot less women, the ratio is much different. It would be interesting to see the differences in behaviors between women where there are more women and then where there are less. So I think, I get pushback from people talking about this, any type of gender, race thing. And I just, to me, science is representative of all of us as people. Let's talk about it. It's fine. I just think people, maybe they feel challenged because there's a bit of guilt there. And it's fine to talk about it. And I like that this was your pick, Cindy, because it gets you thinking about, and my own behavior, I just to not be reserved during question time. If you think like you're going to, embarrass yourself, just go for it. Ask the question, because then you're representative of maybe, helping another woman ask a question, for instance.
So I think that last thing you said is really interesting, because we did, and I don't want to go too much over, but we did an experiment in our seminar series with graduate students. And what we did was, at the end of the talk, we said, okay, we're going to take 5 minutes and everybody think of 3 questions. And then we said, okay, we're going to take 5 minutes and you're going to talk with your neighbor about their questions and your questions. And then we got together as a group, and what became incredibly obvious was that there were so many people with the same question.
Oh, that's cool. Yeah, right.
And it was broader than one would imagine, and it was the same question over and over and over. And so when you think you have a question, you're like, oh, I don't know, because everybody else must know this, and I must just have missed it. There is a very high chance that at least one other person in that room is thinking the same thing. And so I would say, if you have that gut feeling like, I have this question, but I'm not sure, just do it. Because almost never are you going to have a question where somebody's going to say, well, that's a stupid question. I don't know why you even bothered to ask that. The old adage, the only stupid questions are the ones that are not asked.
Yep, right? For sure. Yeah, we have a seminar series like everyone else does, and we have a required course for first year grad students. They have to register and they have to ask a question at every seminar, which is once a week. And there's usually, 5 or 6 new students. So someone takes attendance and they write down that you've asked a question, and that's how you get through the course. But it sounds kind of tough, but they all ask questions and they get used to it. And they realize after a few weeks, oh, this is not so bad, and he or she actually answered my question, and so you get confidence. I think that's a good thing to do also.
I agree.
Start at the very beginning. That's very cool. All right, my pick is a New York Times article from I think it was a week or 2 ago. It's called it's an opinion piece by Nicholas Kristof. It's called Trump's Legacy, Damaged Brains. This is what a common pesticide does to a child's brain. They have an image of a brain which was done here at Columbia University, which indicates where a child's brain is altered after exposure to a drug called chlorpyrifos, which is made by Dow Chemical. It is a nasty chemical. It's a 2012 study found that it was in the umbilical cord blood of 87% of newborn babies tested. It's found in food. It was developed as a nerve gas in Nazi Germany. It's found in food, air, and drinking water. Damages the brain, reduces IQs, causes tremors among children. And now the Trump administration is embracing it, overturning a planned ban that had been in the works for many years. So this is just typical of what's going on. By the way, Dow Chemical, the producer of this compound, donated $1 million to Trump's inauguration. So this is not about politics. This is about keeping people healthy and safe.
That's right.
Having this chemical regulated is the right thing to do. Not allow Dow to sell it to make more profits. That's not what we need.
And I think what this also calls us to do is to listen to the experts and the actual scientists who are doing the work. I think there's a pushback against what are experts, and I think that is damaging, like you said, to public safety. So it's definitely in scary, I think, for some of the things we're seeing with the pushback against science.
Yeah, there's a quote in here that says, kids are told to eat fruits and vegetables, but EPA scientists found levels of this pesticide on such foods up to 140 times the limits deemed safe. So we think we're doing something good by eating fruits and vegetables, but if we're not regulating these types of things that are getting into our food system, we're actually slowly poisoning ourselves, and that's very scary.
Quite.
And for those that are listening to this podcast that I've roped in who are fervent Trump supporters, I hope you're still listening. You'll be fine. You can hear other opinions other than yours. I hear them all the time. You're fine. So just keep listening. If I got you hooked in, you'll like the podcast. I just have to throw that in because I've gotten a lot of people to come and listen who are not maybe in the science community, who are big Trump supporters, but we're going to voice our opinions just as you do yours, and you'll be fine to listen to both sides.
No, we're going to give you the science about why this chemical should be regulated. That's all it is.
Absolutely.
Right. And they'll probably email us.
That's fine.
Oh, it's fine. Email.
We want to have a great discussion here.
Absolutely.
For sure. All right. Immune number 2, you can find it at Apple Podcasts, microbe.tv/immune. And please send us your questions, comments. If you have a listener pick, send that in as well. Immune@microbe.tv. And if you like what we do, consider supporting us financially. We'd be eternally grateful. Go to microbe.tv/contribute. We have a number of ways you can help us out. Cindy Leifer is at Cornell University. You can find her on Twitter, Cindy Leifer, one word, CindyLeifer. Thank you, Cindy.
Absolutely, my pleasure.
Steph Langel is at Ohio State University and on Twitter, Stephanie Langel, all one word. Thanks, Steph.
Thank you. Everyone have a great Thanksgiving.
I'm Vincent Racaniello. You can find me at virology.ws, PROFVRR on Twitter. Music on Immune is by Steve Neal. You can find his work at stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.